All right, so lysosomes and peroxisomes, these are also transport vesicles here. So lysosomes, they form at the Golgi apparatus. They contain enzymes, so they lyse, so cut is how I remember that. So lysing means cut, which anything in the cell that has to do with cutting other molecules up is done by an enzyme. So they contain enzymes. This is really useful in digesting nutrients that have been ingested or phagocyzed bacteria. You need to break it up. Lysosomes, that's the job for them. You can digest other organelles inside of the cell itself that are no longer working properly or have been marked for destruction. That's called autophagy. So basically phagocytosis. So autophagy there. It's a normal, healthy function of the cell for this to occur. When it does not occur correctly, either too little or too much, that's when we start having issues. So an example of like a human disease where lysosomes aren't working properly is Tay-Sachs disease, and it's a genetic disease, so it's inherited, where the enzymes located in the lysosome can't break down gangliosides, which is a specific membrane lipid of neurons. So there are examples of human diseases where simple processes like lysosomes are affected. There are peroxisomes. They are smaller than lysosomes, but they also contain enzymes, but they are mainly breaking down like fatty acids and amino acids. They also have hydrogen peroxide in them because it's a byproduct of that reaction, so breaking those things down. So that's why they're called peroxisomes, is because they have hydrogen peroxide. Funnily enough, there's another enzyme that catalyzes the breakdown of that byproduct hydrogen peroxide into water and oxygen, because hydrogen peroxide is H2O2, right? So it has a similar function, slightly different than lysosomes, but we just call them peroxisomes because they're slightly different. Now, moving on to the next organelle here, mitochondria, the powerhouse of the cell, right? It's the main site of ATP synthesis because you have many cristae or inner foldings of the membrane, the plasma membrane, because inside that membrane you need enzymes and other proteins to really capture and collect energy to make ATP. Because this is a site where electron transport chain is occurring. You probably will learn that in your class with Professor Budovich. But the mitochondria is where this is all occurring. They have their own DNA, basically own genome, which is unique for this organelle. You have so many cristae or inner foldings because you need a large surface area to produce all that energy. When you need lots of energy for the cell, more mitochondria are made or produced. So like muscle cells have lots of mitochondria. So that's a fun little thing. And then there's the electron transmission image here of what that looks like. So it's pretty jam-packed full of mitochondria in that image there. All right, so let's talk about the nucleus here. So the nucleus is the largest membrane structure in the cell. It contains DNA, not DA. That's a typo on my end. It contains the DNA, which the amount of DNA you have in your body is meters long, right? So it's very, very long, and you have to pack that up into the nucleus. So the nucleus has its own liquid suspension called the nucleoplasm that's engulfed and enclosed by the nuclear envelope, which is a double phospholipid membrane layer. So it's 4 phospholipid layers. Way that things move in and out of the nucleus is through nuclear pores. These are proteins that regulate what's coming in and what's going out, because you do not want your DNA floating out, and you don't want proteins or enzymes floating in that could damage your DNA, because then the cell would die. So those nuclear pores are highly important and regulate everything going in and out of the cell. RNA is produced inside the nucleus. When in the next section here, probably in the next video, I'll talk about what this is. But DNA is the genomic information that produces and encodes RNA, which is like a copy of the DNA, which is the nucleic acid, right? It's that intermediary step, which then is that RNA is transported out of the nucleus into either the cytoplasm ribosomes or the rough ER ribosomes to produce proteins. Okay. There is this special region called the nucleolus. This is where it's a dense, pretty dense region, and where a lot of ribosomes are manufactured inside the nucleus, which we're not going to worry too much about on that structure. All right, I will stop the lecture there, and it'll be a shorter video because actually we'll do one more slide here, and then we'll stop. So chromosomes, this is the DNA found in the nucleus, right? So chromosome structure is basically your DNA is tightly packed up in a 3D structure that is composed of a protein called histones and then DNA, right? These histone proteins in DNA, when they're twisted around each other and combined, they form what's called nucleosomes, and they actually help pack the DNA tightly into one another, right? Because you have your DNA nucleic acid strand that wraps around 8 histone proteins, which then these histone proteins, now called nucleosomes when they're combined with the DNA, can twist upon themselves to make chromosomes, like such, which then can fit into the cell. Now, chromosomes are only formed during cell division, mitosis, meiosis. When the cell is not in mitosis, meiosis, chromosomes are not packaged like this. They're just tightly packaged into what's called chromatin, but they're not tightly packed in the chromosomes like we see here, okay? So that's kind of a misnomer that, I thought that when I was a freshman myself at ORU, but this is a rare case of, DNA chromosome structure. This is only found during replication of the cell. All right, now we'll stop there because we'll move into actual gene expression, which can be quite complicated the first time you hear it. So I'll see you in the next video.